Hello, everyone. I'm Sarah Schumacher, and I was one of the moderators for the second session of the Early Career Day, next big thing in cardiovascular research. And today we're just going to talk a little bit about what this program is for and highlight some of the exciting new science that we saw today. So how this works is the next best thing in cardiovascular research was based on all of the early career abstracts that were submitted to the BCVS program. And these were scored by faculty, and the top scoring abstracts were selected to present an oral talk today during the sessions.
Hello, everyone. My name is Patricia Nugyen. I'm an assistant professor at Stanford University, and I was a moderator for session 1, the next best thing in cardiovascular research in the early career session. So I wanted to highlight 2 amazing studies that were presented by our early career investigators. The first is pharmacological inhibition of macrophage infiltration prevents myxomatous valve degeneration in Marfan syndrome by Na Zhu of the Cincinnati Children's Hospital Medical Center. And the other study is time of intake regulates glucocorticoid pharmacology of cardiac bioenergetics by Mattia Quattroselli of also the University of Cincinnati. So I wanted to say how amazing these studies were because they highlight the importance and the emerging risk factor that inflammation is for cardiac disease. And the first study evaluated a inhibitor of CCR2, which is a macrophage chemokine that attracts macrophages to the area. And Na Zhu presented very interesting data on how inhibiting macrophage infiltration can actually reverse the disease in a mouse model of Marfan syndrome. So this is very exciting, and at some point in the future may become a translatable treatment for myxomatous mitral valve disease, which is a very prevalent disease that we currently do not have treatment for. And the other study that was presented was also a potential treatment. Mattia Quattroselli presented on the potential for glucocorticoid therapy to treat diabetics and aging hearts that have abnormal bioenergetics. And the interesting thing is that glucocorticoids have been with us for a long time, and the value of them, apparently, according to this study, is basically the timing of administration of the glucocorticoid therapy. So those are 2 amazing talks, and now I'm going to turn it over to Dr. Bopassa to review the other 2 talks in the session.
I'm Chrisostome Bopassa from UT Health Science Center in San Antonio. I moderated the session with these 2 studies. The first one from Emily Flam, coming from the University of Pennsylvania. He presented her work entitled Integrated Cardiac Metabolism in End-Stage Human Heart Failure. The author of this work, this fantastic work, provided a comprehensive depiction of cardiac and systemic metabolic changes in 89 explanted failing and non-failing human heart through integration of plasma and cardiac tissue, metabolomics, genomic, wide RNA-seq, and proteomic data. This was a very, very fishing expedition from this group that compared the failing heart versus the non-failing heart, and they discovered many bioenergetic factors that are associating with the heart failure. The problem of the study was we did not know if whether what they found as factors were the cause of the failing heart or the consequence. In summary, it is a very, very nice study that pointed many factors, including fatty acid, TCA cycle, intermediates, and ketones. This was really a very good work that opened my eyes in the way to see the next stage of heart failure studies. The second study that we heard this morning came from MeowQ. Using a spatial transcriptomic profiling, MeowQ and collaborator visualize the regenerative cardiomyocyte reconstituting damaged myocardium after ischemia. And the good thing was they found that the expression level of NRF1 and ER band stress responsive transcription factor was regulated. So the key finding of this group was that the cardioprotective function of NRF1 is mediated by a dual stress response mechanism involving activation of the proteasome and maintenance of the redox balance. This was very important because it uncovered a unique adaptive mechanism activated in response to injury that will maintain the tissue homeostatic balance required for the heart regeneration. I did like this because, you know, the regeneration of the heart tissue is a very complicated field with a lot of discrepancies. This group has pointed the role of NRF1 in the regeneration of the heart. I believe the next step will be to work on to try to see whether manipulation of NRF1 will help to regenerate the tissue. We all know that after ischemia, by example, the tissue death is the tissue injury is the problem. Regenerating this tissue, finding the way to regenerate this tissue is one of the big fields that we all are expecting to grow. I believe that the work from MeowQ was really clear and help us to understand new mechanism in tissue regeneration. I would like to thank you all for attending this two sessions that were very nice to follow. Great talks, and really we thank the presenter, the speakers for taking in time to put everything together to help us to understand their studies.
So I want to echo Dr. Bopassa's comments that these were amazing presentations that are showing great science, and we look forward to the next steps in your studies. And now would like to turn the sessions to the next moderators who will review the next big thing in cardiovascular research session 2. Thank you so much.
And today I'll highlight 2 of the speakers, and my colleague Mo will handle the other 2. So our first speaker was Abraham Beyer, and Abraham was talking about the role of Th1 cells, and particularly MyD88 in Th1 cells, as far as hypertrophy and fibrosis in a model of transaortic constriction. And so what he found was that MyD88 knockout is actually very relevant to Th1 infiltration and the mediation of fibrosis and activation of myofibroblasts. And so it's, as people know, the hypertrophic response is partially adaptive, but also very maladaptive. And so understanding how immune cells are being called into the heart and what they're doing once they're in the heart, and how we can maybe moderate that response to be more adaptive and less maladaptive is very important. So understanding Th1's role and what's causing their activation and maintenance in the heart is very critical for developing future therapies. And the second speaker I will cover is Nathan Van Dusen. And so Nathan was talking about CRISPR-Cas9 genome editing technology. And so he's using a new adeno-associated viral method for homology-directed repair in order to cause more precise genome editing in the heart or designer tissues or designated tissues. And so this is a method to increase the efficiency and proficiency with which we edit the genome. And this would be for gene-based therapies for disease. So especially diseases where there's a early deletion or an insertion that causes aberrant transcription and translation of a protein in the heart. And so this is very exciting as we think towards either patient-specific therapies where there's a specific mutation causing disease, or in general where there are truncated proteins where we need gene editing in order to allow the full length to be produced and thus the full function to be performed within the heart. And so these are both very important and very relevant talks for the future of cardiovascular research and therapy. And in general, it was a great session. It was well attended, and there was a very active Q&A. And so we really appreciate this involvement in the early career events. This is wonderful for them getting known in both the BCVS and general, the cardiovascular research field, but also for them to get feedback on their science. So at this point, I'll turn it over to my colleague Mo to talk about the other two talks in our session. Thank you.
Thank you so much, Sarah, for that. So yeah, my name is Mo Al-khalaf. I'm a research fellow at the University of Ottawa Heart Institute, and I co-moderated with Sarah the second early career session titled The Next Best Thing in Cardiovascular Research. So, for the talk titled Novel Functions for the Synaptosomal Associated Protein 29, or SNAP29, in Cardiac Arrhythmias, speaker Dr. Ji Wang from the University of California, San Diego, shared their study results that uncover a novel role for a protein named SNAP29, which functions in the regulation of autophagy, or cellular content self-eating mechanism, which happens normally as cells recycle and clean up internal proteins that degrade or otherwise need to be disposed of. Now, their study shows that SNAP29 insulated a subset of desmosomal and gap junction proteins from selective autophagy-mediated degradation to restrict cardiac arrhythmias. So the novel thing here that was shared with us is that the loss of the SNAP29 desmosome gap junction interactome may predispose the heart to desmosomal-based diseases of electrical nature, such as atrial fibrillations and other types of heart disease arrhythmias. Now, for the other talk that was presented in the session, the title being the selective IGF-1 production by resident cardiac macrophages orchestrates adaptive cardiomyocyte growth during hypertensive stress. This was presented by PhD candidate Raisa Zaman from the University of Toronto in Ontario, Canada. And what makes this exciting is that the study has been just recently published and made public in the high-impact journal Immunity just a couple of weeks ago. In this presentation, Raisa defines the absolute requirement of cardiac resident macrophage-produced IGF-1 in adaptive cardiomyocyte growth during hypertension, identifying a novel and essential pathway for resident macrophage-directed cardiac adaptation to hypertensive disease.
So thank you again from Mo and I for attending the Next Big Thing Session 2. Today we had speakers from all over North America, from Tufts University, UC San Diego, Boston Children's, and the University of Toronto. And this opportunity, the Next Big Thing sessions, are really an opportunity to highlight up-and-coming early careers, give them a chance to voice their science, get feedback, and really start to make a name for themselves and start to network within the cardiovascular research community. So this is a great opportunity, and we're very much a fan of promoting the training and engagement of early careers. And if you have any questions regarding that, feel free to reach out to Mo or myself or any of the early career committee so that we can help you as you take your own path in cardiovascular research.